notes, they're updated versions, so you can be downloaded more than an hour ago. You can download. If you did print out invitation notes for everyone, look for a little red dot in the lower right-hand corner. That will tell you that's a modified slide. Missing a location. So that's going to show it. So if you can download it, do so. Not no big deal. There's a couple slides and then a little red dot. Outside of that, Gary has a quick announcement.
There we go. That raises awareness and money for affordable housing, and it works with other ones like Habitat for Humanity, and Rebuilding Together. So we bike across the country, we stop along the way to fix houses and everything like that. I highly encourage you to check it out just because it's an awesome program and a great way to spend the summer. But in addition to that, so for my fundraising, Panda Express has agreed to donate 20% of their profits today to me if you mention Carrie and Bike and Build and hand them one of these flyers, or tell them you forgot the flyer and insist that you know what it's about. And also, I will be hosting a 5K on April 1st, which is Fool's Day at 1 p.m. It's a Sunday. And the catch is, it's not the winner it's not the fastest person that's going to win, it's the person that predicts their time the closest. So it's around the Alt-Bueller Trail, which I'm sure all of you guys have done at least once. Obviously obviously no GPSs and your fancy watches and all that sort of thing. But it should be a lot of fun. I've gotten a ton of food trucks to come, and we have some great prizes to give out. So I highly encourage you to come out. It's a great cause. It's a great time. So yeah, eat a pan today and then come run it off on April Fool's Day. So thank you.
All right. For those of you just wandering in, there's a couple changes between the versions of the PowerPoint and the PDF that were up there most of the night. If you downloaded more than an hour ago, you might want to re-download, or just watch for the little red dots that are going to appear right here. That'll tell you where the modified slides are. They're not a lot of big changes, but just so you know. Okay, so we've been over how transcription works, how the enzyme functions, how it starts, how it elongates, how it terminates. We talked a little bit about the logic of transcriptional regulation. What we're going to do this week is talk about some examples where transcriptional regulation has been worked out in quite a bit of detail. We want you to think about applying the ideas that we went over last week to some specific cases. And today I'm going to talk about prokaryotic transcriptional regulation, and on Wednesday Steve's going to talk about eukaryotic transcriptional regulation. So the first thing I want to do briefly at the beginning is just talk about two particular features of transcriptional control in prokaryotes that are unique. And they're the presence of operons and transcriptional coupling. There are operons outside of prokaryotes. In fact, there are eukaryotic operons, but for the most part, they're an exception, but they're a big part of the way transcription occurs in prokaryotes, in particular eubacteria. So I'm just going to say a little bit about those. And then the rest of the time, we're going to go over three examples: the lac operon, the lambda switch, and ribosomal gene operon. And I want you to be thinking about how the logic of regulation works. We talked about these as being analogous to switches, that you can turn genes on and off, you can manipulate their level. How does the cell execute that logic? And then secondarily, or in addition to that, what are the molecular mechanisms that actually execute that logic? So I want you to think about these on two levels. What's going on? What's the cell trying to accomplish here? And how does it do it? Okay, so those are the two things. So three examples, and think about both of those kinds of issues for each example. Okay, so first, some unique features in eubacteria. The first is the presence of operons. The defining feature of operons is that you're going to make multiple proteins from one messenger RNA. multiple proteins from one messenger RNA. In your body, or your genome, there are 22,000 odd-ish number of genes that make proteins. You sometimes make more than one protein from a single messenger RNA, but you do it by changing the splicing. Here we're talking about completely unrelated genes that are part of one giant messenger RNA. So this is an example, tryptophan operon. There's one promoter, the polymerase attaches here, and it goes across 5 different genes, and there's one terminator. So you make one giant messenger RNA, and out of that, you're making 5 separate proteins. These are completely unrelated to each other in terms of their structure, but they are harmonious or related to each other in terms of their function because they execute a single pathway. And that pathway is to synthesize an amino acid, tryptophan. So this is a starting point, chorismic acid, and these proteins are the steps in making tryptophan out of chorismic acid. So when you run out of tryptophan, you want to turn on this operon, and it's all the parts you need to make that one essential compound. Okay. So they're not random genes that are put together in operons, they're genes that work together. So the 2 essential things I want you to know about operons is that you're making multiple structurally unrelated proteins out of a single messenger RNA, but those proteins, even though they're structurally unrelated, function together to execute some logical set of processes that you would need at the same time. You're never going to need this step unless you also have these guys. So that's how the bacteria regulates. Now in your body, you have biochemical pathways too, but genes tend to be regulated independently. Okay, so that's a big difference between eubacteria and eukaryotes. Any questions about operons? The other thing is that there's no nucleus. And so translation can begin as soon as transcription starts. And you can visualize that in this electron micrograph, where you can see, actually, this is a strand of DNA here. And you're actually getting these little dots are ribosomes, and these strings are messenger RNA molecules. So this was a method of visualizing transcription that was worked out by Oscar Miller and Barbara Beatty published in 1970, the late 60s. And they showed definitively that you're having simultaneous transcription and translation going on. So the next slide kind of deconstructs this and shows you what the parts are. So this is a strand of DNA. This would be an RNA molecule. That's RNA polymerase. You can see a tiny little dot at the base. It's smaller than a ribosome. The ribosomes are the big dots. What are 2 things you can tell me about transcription that come out of just seeing this picture? Yeah. Right, so this is a really important point. When you think about transcriptional regulation, don't think about attaching RNA polymerase and making a messenger RNA and then we're done. This is a dynamic process, and there are multiple RNA polymerases going across the gene at the same time. And look how many proteins are being made from each mRNA. So there's a multiplication process here. When you turn on a gene, you're making a lot of mRNA, and out of every mRNA molecule, you're making a lot of proteins, a lot of individual proteins. Yeah. You can't actually see the protein. So any questions about what this is showing you? Well, let me just ask a quick question. Where's the start site of transcription on here? Where would it be? Would it be over here or over here? On which side, the right or the left side? The left side, and how do you know? Well, I wrote 3' there, but how would you know just looking at the picture? You should be able to figure this out. Yeah. Absolutely correct. But mRNA molecules are long over here and they're short over here. So as the polymerase moves along, it's making a longer and longer mRNA, right? So you're visualizing, this is a very, I know it's static in the sense it's one photograph, but it's capturing a dynamic process. There's a lot you can learn from looking at something like this. Okay, so this is called transcription, translation, coupling. Just one second. Because the two processes are happening at the same time. This cannot happen in a eukaryotic cell because the ribosomes are in the cytoplasm and transcription is happening in the nucleus. But in a bacterium, pretty much as a messenger RNA molecule emerges, translation can begin. Yeah, question. Yeah, here's some short ones, medium, and here's some long ones. So as the polymerase moves across, the RNA is getting longer and longer and longer, and then it falls off. And then this is probably the beginning of the next gene. And you have a short one again. Because when it starts, it's just one nucleotide, and when you end, it's hundreds. So it's getting as the polymerase moves along, it's adding more and more nucleotides, so the chain's getting longer and longer.
Wait. Does this make sense?
Because you have multiple polymerases going across at once.
Shit. Oh, never mind.
All of these are, that's just one example. And these are a few ribosomes, but there are many. So that's an mRNA molecule, that's an mRNA molecule, that's an mRNA molecule, and that's an mRNA. So there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, at least 15 messenger RNAs. That means there's 15 RNA polymerases on the same gene at the same time. Each one's making a messenger RNA. So you're loading one, and as soon as it moves off the promoter, you can add a second one, and as soon as it moves off the promoter, a third one, and so you're shunting them through the gene. Before the first one falls off, you've got other ones coming in behind. Yeah. Yeah, every mRNA is going to have the same sequence because it's the same gene. So they're using the same template strand. Yeah. Because the short molecules are on this end and the long molecules are on this end. So when the polymerase starts, it's adding one nucleotide, and then a second, and then a third. So the chain's getting longer as it comes out the back of the polymerase. Remember how it exits? And so during elongation, the RNA molecule is getting longer and longer. So this is the short, it's short here and long here, so this is the direction. Well, they're short here because it hasn't had time to add them. These ones will get long and they'll move over here. They just haven't gotten to it. So this one, so the beginning, the promoter is probably somewhere around here. Maybe it's actually there, I don't know. That might be another gene, but got it now? Okay. Any other questions about how to interpret this picture? So even though it looks really simple when you first see it, you can learn a lot just from thinking about what's actually going on here. And if you had a lot of these, you could actually get some numbers in terms of, on average, how many proteins made from each gene, from each mRNA, on average, how many polymerases per gene, and that would give you an estimate of the dynamic properties in a living cell. Okay. Good. Now let's apply what you just have learned. Imagine these two, here's the micrograph we were just looking at, and here's another micrograph of the same kind. Okay, it's showing you basically the same thing. Assume that the distance from here to here is the same as the distance from here to here. Which gene is being transcribed at a higher rate? Okay, we probably don't even really need to stop, drop, and roll here. Just think about this for a minute. And who's got an answer? Come on. I know it's Monday morning. It's Monday morning for me too. Yeah. The left one, and why? More strands. So these are all RNA molecules coming up. Now it actually turns out this is a ribosomal gene, not a messenger RNA. It's not a protein coding gene. So there are no ribosomes on there because it's not going to be made into protein. But these are RNA molecules, and there's a polymerase at the bottom of every one of these. So you have a high density versus a low density. That means more RNAs, molecules, in the same amount of time. It's being transcribed at a higher rate. Yeah. Correct. They're in the same orientation. The start site's right there. So really short, longer, 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 and then they fall off. And because there's so many, you can see the exact extent of the gene. Here, it's not quite, I don't know whether the promoter's there, but you're past it here. I don't know where the terminator is exactly, but it's probably somewhere in there. But here you can see exactly the length of the gene. Yeah. Right. So the question is, this is going to become a ribosome because it's ribosomal RNA that's being made. These get chopped up into pieces and then they associate with protein, so they're folding during that process. Right now, they're spread out. Now, I should say the other thing is that the process of preparing the DNA and RNA for these pictures involves using a detergent, so that's going to unfold them even if they're folded in the cell. Okay, so the question was about the folding of the RNA, because remember, RNAs start to fold into hairpins and clover leaves and structures like that. In the cell, it wouldn't necessarily look quite this spread out because that's just the way it's prepared for the micrograph, using a detergent to spread it. So it could well be folded inside the cell. Okay, any questions about operons or transcription translation coupling and how we visualize these images, how we think about them? Any questions about that? All right, the first of the three examples I want to cover today is the black operon. So you should be hearing operon, you should be thinking, okay, we're going to be talking about control of multiple proteins at once through the production of one mRNA. Okay, that's what an operon is. And the black operon is important historically because it was the first ever demonstration that there are non-coding sequences that do something. Okay. And that work was done in the early 60s, and the Nobel Prize in Physiology and Medicine was awarded in 1965 to Francois Jacob and Jacques Monod for doing this work. So it's a really famous example. It's been extremely well studied. The recognition grew out of an observation. Like so many discoveries in science, they weren't actually looking for what they found. They were looking for something else and observed something that piqued their interest and followed up, and that's what happened. So the observation was the following. If you grow E. coli cells in the presence of two different sugars, it looks like it would prefer to use one and then switch to using the other. And this was discovered by Jacques Monod. He called it dioxin growth, means two growth phases. And it's actually easier to see down in this bottom row where you have a really rapid period of growth, and then things kind of stop for a while, and then you have another rapid period of growth. And so you've got this little hiccup in the middle. And the interpretation he had is that you first use up the glucose. The cells prefer to use glucose as an energy source. And once they've completely depleted glucose, they switch to using the second sugar. So dioxit means two, and AUX comes from the Latin word for growth. Okay, so that was his interpretation, but that's not a molecular mechanism, that's just an observation. So how does the cell actually execute that regulation? Well, they posited that cells could actually respond to the presence of small molecules in the environment, in this case glucose, and change their gene expression in response to the presence of glucose, or in this case lactose. And the way you can do that, we can now show, is if you add lactose to a set of cells that have no sugar at all, they'll start to make messenger RNA for lactose. And they'll start producing protein. These two colored lines are protein levels. So they'll make a protein that's an enzyme that digests the lactose, and they'll make a transport molecule in the membrane to get more lactose inside the cell. So they're responding to the presence of lactose in their environment. We call it inducible because it means you're turning on the gene, you're inducing its expression in response to something in the environment. This is a very, very useful thing for a cell to be able to do, but at the time it was first discovered, nobody understood how it actually worked. So that's what we're going to talk about, how this inducible gene expression happens. We now know quite a lot about the structure of the lac operon. And it basically looks like this. There are 3 protein coding genes. There's a promoter where the RNA polymerase is going to bind. And between the promoter and the operator, Jacob and Monod proposed that there's another non-coding piece of DNA that they call the operator. It was essential for the correct regulation of this operon. There's a 4th gene that has its own promoter called LAC-I, and it makes something called a repressor protein. So this is the organization of the lac operon. These are the 3, one's actually a transport molecule, the other 2 are enzymes that are going to utilize lactose when it's available. Okay, so this is an animation, so I could run it more than once, but just stop writing for a second and I'll walk you through how this works. So the LAC-I gene is going to make a protein called the lac repressor, and it's the key to keeping the gene, as the name implies, keeping this operon off. So it's a repressor protein. It's a transcription factor whose job is to repress. Okay. So it goes over and it binds to the operator region. Now, when it's bound, what would happen if RNA polymerase attached here? It could not transit across and start making an mRNA. It's physically blocking it. So that's one way a repressor can work. You just physically block the polymerase, and that's all it's doing here. So its DNA binding domain recognizes a sequence in the operator region. Right? Now, when the cell has lactose in the medium, it goes over and attaches to the repressor. I'm going to play this again, so just don't worry about it. It attaches to the repressor, which then changes its conformation and pops off the operator. That means that the polymerase can attach. It makes a single RNA molecule. Those get turned into 3 different proteins. And now the cell can utilize that sugar for energy, and it can grow. Okay, so shall I do it again? Or is that okay? So this is always going to be made. This protein's always being made by the cell. What it does depends on whether the cell is in the presence of lactose or not. Okay? So when there's no lactose present, it binds and it represses. When lactose comes inside the cell, it binds to the other end of the molecule, induces a conformational change, it pops off the operator, polymerase transits the operon, you get the production of the 3 proteins, and they can utilize this as an energy source, and the cell grows and it's happy. Yeah?
So it's not that when there's no lactose, it's repressed. It's just perpetually repressed until the introduction of lactose?
Right, so this is a really good point, and I'm going to sort of highlight that in just a few minutes, but what you've noticed is that this operon's default state is off. Okay, that's the logic of this operon. The default state is off. It's called an inducible operon because the change is to turn it on. The trip operon, which I showed you at the beginning's default state is on, and you can turn. So, yeah.
If you look at this slide and slide before it, and the previous slide, the repressor protein goes to the promoter, or does it go to the operator?
Yeah, that arrow should be to the operator. Yeah. They're, as I'll show you in just a second, they're essentially touching each other. Okay, so one more time, because you have to go by this now. Sugar changes conformation, releases the operator, polymerase binds to the promoter, makes an mRNA. Translation happens now, you know, immediately. And you have 3 different proteins. They allow you to utilize that energy source, and the cell grows. Okay, so this is the default state for the operon to be off. So it's the repressed state. When no lactose is present, the repressor protein binds to the operator region. As you can see, they're right next to each other. This keeps the polymerase from attaching. See, it keeps trying and it keeps falling off. It's very sad. It can't do what it needs to. And so there's no messenger RNA produced. Okay. So that's the default state. This is what the promoter and operator regions look like. This is where the lac repressor binds in the operator. The promoter is right over there. So it'd be -10, -35 would be just a little upstream. And you can see it's actually a mirror image binding site. That's kind of a clue about the nature of the protein that's going to bind there, which we'll get to in just a second. So it's between the start of transcription and where the gene would go. So that's why that's the operator promoter boundary, literally right there. Now, one of the reasons we know that the operator functions correctly, it does this job, is that if there are mutations that change this sequence in here, all of these separate mutations take you away from the consensus, which means that the repressor protein can't bind as well. And what that means is that this operon is going to be doing what all the time? It'll be on. It can't be repressed very well if the repressor protein can't bind. So these mutations all change it from being inducible to a little bit at least on all the time. And that's one of the lines of evidence as to how this whole system functions. This is the structure of the lac repressor. It's a homotetramer. So 4 of the same thing make a structure that binds. As with all transcription factors, there's a DNA binding domain, and then there's a protein-protein interaction domain up here. So this part up here makes sure that the 4 molecules all join together, and then it's really just physically blocking the interaction of polymerase or the transit of polymerase across the DNA. So it doesn't really have to do anything else except get in the way. So the essential parts of this molecule are the DNA binding domains and the tetramerization domain. So that's kind of a close-up. That's another picture of it. Now let's have one other piece that's going to be important, and that's where the ligand is going to bind to change the conformation. Okay, we'll get to that in just a second. Everybody cool? Now when the ligand binds in that pocket right in there, it's going to go from being attached to the DNA to letting go. Conformational change, crucial to many, many aspects of transcriptional regulation, many aspects of cell signaling, lots of different ways to change the activity of the protein in many different contexts through conformational changes. Yeah. Right. Thank you, Steve. A ligand is a small molecule that attaches to a protein. So in this case, the early idea was that that ligand is actually lactose. Turns out it's a little bit more complicated. I'll get to that in just a second. But the early models for how this was functioning was that it was actually lactose itself that's binding to the lac repressor and causing it to change conformation and pop off the DNA. That was what we thought for a long time. It turns out subtly different, but to all intents and purposes, the same idea. A small molecule is binding, and that's inducing the conformation change. So that molecule, so one way to think about this is it's a repressor, but it's also an environmental sensor. If that small molecule is present, that ligand's present, it will bind and cause the protein to change shape. So it has both of those functions. It's acting as a repressor, but also a sensor of the environment. And that's what allows it to throw the switch to allowing transcription to happen. Okay, so the induced state, and you see in this picture it's actually drawn as lactose binding and popping it off. We actually know now that it's not that molecule, but I'll get to that in a second. But anyway, if a small molecule binds to the repressor, it can no longer attach to the operator region. So now the polymerase can attach to the promoter, and it goes across the gene, and it makes the messenger RNA, and you get the proteins that are necessary for utilizing lactose as an energy source. That's the essential switch. Now, it's actually a little bit more complicated in a couple ways, and we need to go through this because there's some additional things that make it really interesting and actually make it work a lot better than this. Okay, you actually have the piece of evidence that led us to understand a little bit more about how this works. Even though lactose is necessary to induce transcription of the lacto operon, we actually already know that it's not sufficient for high levels of expression. And how do we know that? I actually showed you a slide earlier today. I know it's Monday morning. I actually showed you a slide earlier today that provides experimental evidence that just having lactose in the cell is not going to be enough to give you transcription of this, or at least very much transcription of this operon. Well, so take care, Sharon. Or do you have a question before we get to that? All right. What do you think is the evidence that lactose alone is not going to turn this operon on?
Like that. Are you actually? I could. I like her. Let me use my girlfriend in
It's switch. It's that with like maybe in the absence of glucose, you try to use the lactose. Maybe
All right. Who has an answer for us? Hard question, huh?
Well, we talked about before with the dioxic growth that, like, it's only going to metabolize lactose when there's no glucose in the cell. So not only do you have to have lactose present, you have to have no glucose.
Exactly right. Perfect. Wow, 4 points. Awesome. All right. So the answer is that the dioxic growth shows you that it's not actually ready to metabolize lactose until the glucose is gone. Otherwise, it wouldn't be that pause. It would just go straight into utilizing the lactose. But there's instead that little pause, the dioxic growth curve, is telling you that it first uses up the glucose, and it's not until the glucose is absent that the system really kicks into gear. So we now need to understand the molecular mechanism that executes that change. And that involves a second protein. So, okay, here's the dioxic growth. So the cells are growing very happily. They use up the glucose. They kind of wait a little while, and then they get going again as they start to use the lactose. That's the black line. So that's the number of cells. On this axis is the expression of beta-galactosidase. So if you actually measure one of the enzymes that comes out of that operon, which is beta-galactosidase, there's really none produced until you get to this plateau, and then you start to make a lot of it. So something is kicking on a higher level of expression, or actually really turning on this operon at a substantial level after all the glucose is depleted. So how does that work? So here's just a summary, a different way of saying the same thing. When glucose is present, the lac operon's off because you don't have any lactose. But even if you have lactose and glucose, the lac operon's off Well, not really off. We'll get to that in a second. It's only when you have lactose alone that you truly have this operant cranking out a lot of messenger RNA and protein. Okay? And the protein that is involved in mediating that switch is called CAP or CRP. So this is one of the slides that changed because last year the textbook we were using called it CRP and your textbook calls it CAP, so I switched those around. So that's the big change on this slide. So when glucose is not being utilized, in other words, when there's no more glucose left for the cells to grow on, a small molecule called cyclic AMP that's used in signaling starts to increase in concentration. And cyclic AMP is a ligand for CAP. Okay, so remember, a ligand is a small molecule that binds to a protein and changes its conformation. So cyclic AMP levels start to rise when there's no more glucose. It binds to CAP, and CAP is a transcription factor that can activate expression of about 100 genes, and one of those is, one of its binding sites is just upstream of the lac operant. Okay? So here's the promoter region, so the operator is right there. Here's the promoter with the -10 and -35 sites, and upstream of that is a binding site for CRP, CAP. And it binds there, and it helps through this. This is the protein DNA binding domain is down here, and the protein-protein interaction domain is there. They interact with the alpha subunit of RNA polymerase and help stabilize that interaction. And when that happens, expression levels go way up. Okay? So one way to think about this is it's a two-component switch. The lac repressor is an environmental sensor and a repressor that gets in the way of polymerase making mRNA. If it's bound to the DNA, you basically can't make mRNA. If you get rid of it, there's a small amount of transcription that happens because RNA polymerase can attach on its own. But it only makes a little bit of messenger RNA. But when the cyclic AMP levels rise after glucose depletion, then you have CAP, CRP binding, and that really attaches the polymerase well, and you get a lot of transcription. Okay, so it's a two-component switch. So any questions about how that works? The role of CAP in activating expression. So getting rid of the repressor isn't really enough to give you high levels of transcription. They're very low levels, basal levels of transcription. It's the addition of CAP that brings down the polymerase and really kicks that promoter into high gear. Okay. Now, I told you that in the old days we used to think lactose was the ligand. This is a slightly modified slide too, but it's not much changed. The text is pretty much all the same. It's just a better picture. We used to think lactose was a ligand for the lac repressor protein. It's actually a related disaccharide called allolactose. It turns out that the beta-galactosidase enzyme, which breaks down lactose into the two-component sugars that then get used for energy, it mostly catalyzes that reaction. So this is the energy source broken down, and then this goes straight into glycolysis, and this gets changed into something that becomes glucose that goes straight into glycolysis. So this all gets burnt for energy. But a little bit of allolactose is produced, and this is actually the ligand of the lac repressor. It's a small detail, but it's not actually lactose that's binding to the repressor protein. It's actually allolactose. That's all you really need to know about allolactose. It's the true ligand of the lac repressor. All right, I threw this extra slide in just because I wanted to tie in with what you guys are doing in the lab. So you're using blue light screening to see whether you have inserts in your bacterial colonies. And there's two components that you put into the medium that are utilizing beta-galactosidase. And this actually the whole lac operant. Okay. One of those is XGAL, which is not actually lactose, but it's called a lactose analog, which means that the enzyme can recognize it and cleave it. It's not what the enzyme, in a sense, is designed to work on. It's an artificial molecule that the cell doesn't normally see. But it can cleave that linkage right there. We don't care about this anymore. That's an indole ring that can turn into a brightly colored compound. That's blue. The other component that you put into the medium that's part of this system is IPTG, which is a ligand for the lactopressor protein. So instead of allolactose binding, IPTG is going to bind to the, and it's a very strong ligand. It doesn't pop off again. So what it means is it's going to de-repress the operon continuously. It's going to make sure that all the lactopressor protein is bound with a ligand and is therefore off the DNA. So that's what those two components are in your medium that you use in the lab. Any questions about that? So we're harnessing this system for practical use here. But a normal cell would never really see these two compounds. Any questions about what they're doing? Yeah. So what's happening is the beta-galactosidase, if you don't, so anybody remember, so white means that there's an insert and blue means there's no insert. So if this reaction occurs, you get blue, and that means the beta-galactosidase gene is fine. If you've successfully cloned a piece and you've essentially inserted it into the middle of the gene encoding the enzyme, you can't make the enzyme. Therefore, you can't turn the X-gal, which is colorless, into this blue compound. And that is the basis for how you do the blue-white screening. It's utilizing the beta-galactosidase gene as an indicator. And if you clone successfully, you can't make the enzyme. If you don't have an insert, the enzyme's still there, and you turn X-gal into a blue compound, which you can see, and that's what the blue colonies are. And again, this is a ligand for the lactopressor protein. Make sure that it's always off the DNA so you can get this enzyme produced. Otherwise, you wouldn't have the enzyme produced. That's why you need both components. Questions about the molecular basis for blue-white screening? You should understand this at a pretty good level now. Okay, so this is to summarize what we've been over with regard to the lac operon. This is a picture from your textbook. This is just the general layout of the operon with the promoter and operator. There's the lac repressor gene. There's the site where the cap protein binds, and these are the three genes in the operon. If glucose let's start with it off. If you have no glucose or low glucose and there's no lactose, you're off because the repressor protein binds. If you have a lot of glucose and there's no lactose, it's off because the repressor protein binds. So those are the same state. If lactose is present and you also have glucose, the repressor protein comes off and you get a little bit of expression. But again, you have glucose. So it's just a little bit of binding. It's not that you're activating transcription, you're just no longer blocking it. But if there's no glucose or very low glucose, cyclic AMP levels go up, cyclic AMP binds to cap changes its conformation and binds to its binding site, and then that interacts with these alpha subunits of the polymerase. The polymerase now attaches very well, and you have high levels of expression. Yeah. I know, but it's the one in the book. Yeah, so Steve raises a good point. And it tells you a little bit about the logic of the switch. If you have low glucose and no lactose, both are going to be bound. But in this case, the blockage wins. So what that tells you is that the binding of cap is really only going to activate transcription in the absence of binding by the lac repressor. Okay, so that wins. If both are bound, this wins. But if this guy's gone, then having this guy bound gives you very high level of expression. Yeah. The order of binding, I don't know, but my understanding, I'm pretty sure, is that cap's bound, and it's helping to attach multiple So the cap stays bound and it's bringing down multiple polymerases one after the other. That's my understanding of the way it works. But I don't know for sure. Okay, any questions about lac? Okay, this is just a summary, and this is another slide that changed. And I changed this to reflect binding rather than whether the operon's on or off. I think it makes a lot more sense. So a key to the way this works is the ligands. The ligands are indicating the physiological status of the cell, and so the cell's taking advantage. They're direct indicators of what's going on in the cell. So the switch is essentially taking advantage of knowing, is there lactose present, is there glucose present? And cyclic AMP is an indirect indicator of the level of glucose. So CAP is sensing glucose level indirectly, and lac repressor is sensing the presence or absence of lactose, and between the two of those, it can execute the switch. And so these ligands are providing that crucial information about the environment. Yeah.
So if nobody knows the lac repressor depends on how lactate, that depends on the beta lactate.
Sorry, I'm gonna have to run up there.
So it seems to me you need beta galactosidase to make allolactase in order to make more beta galactosidase.
Right. Okay. So you bring up another inconvenient truth. Right. So the system seems like you need a little beta galactosidase, or you couldn't make allolactose to get the system going, and that's absolutely correct. So the truth is, there's always a little. Remember, we said no gene is ever completely off. There's always at least a couple molecules, like 1 or 2 proteins of beta galactosidase present in the cell, and that's really all it takes to get the system going. So it's never completely off, and you're exactly right. That was a very perceptive question. Okay, so what I want you to think about is lac as an inducible operon, and an example of a repressible operon is the trip operon, which I showed you at the very beginning. I'll show you one more picture in a second. But an inducible operon is by default off, and when the ligand binds, at least in the case of lac, it's going to release the repressor, which allows it to come on. It doesn't turn it on, but it allows it to come on. Remember, CAP is really turning it on. The trip operon, on the other hand, is usually on, but can be turned off. So it's repressible, and it works like this. So normally, most bacterial cells are going to be growing in an environment where there's not tryptophan present. Maybe there's sugar present, but it's going to have to make its own amino acids. And tryptophan is an essential amino acid, so it typically is going to be producing the enzymes that can make tryptophan. But if you start to get a lot of tryptophan in the cell, you don't need to do that anymore. You've got plenty of tryptophan. In those conditions, you want to turn it off to save energy. So tryptophan binds to the repressor, and the repressor then binds to the operator. So that turns it off. Okay? So it's normally on, but when the ligand is present, it binds to the repressor, and the repressor then attaches. Remember, with the lac repressor, when the ligand bound, it let go. So the switch can be executed in the opposite direction, and that makes it repressible rather than inducible. That's really all I want you to know about the trip operon. It just works in the opposite direction. Okay. Any questions about that? Okay, lambda repressor. Another classic system, very, very, very well studied. There's a lot known about this. I really want you to focus on just some pretty basic aspects of how this switch is thrown. Again, it's a switch from one state to another. It works a little bit different way. It just shows that the logic of throwing a switch in regulation can be executed in a different manner. Okay. So we're going to be talking about genes that are part of a bacteriophage or virus genome, and that bacteriophage is called lambda. Here's a electron micrograph of it. Here's a sort of picture of what it might really look like. Lambda is a bacteriophage, which literally means bacteria eater. And it's a parasite of bacterial cells, including E. coli. So we can grow it in E. coli. And when it goes inside, you'll probably remember from the Hershey-Chase experiment that these bacteriophages attach to the cell membrane and they insert their DNA into the cell. Now, in the case of lambda, one of two things can happen. One possibility is that it inserts into the host genome. It actually integrates into the E. coli circular DNA. It cuts it open, sticks itself in, and then ligates. And you should now know what those enzymes are that do that. There's an endonuclease and a ligase. And it can then start to produce more of itself. Of course, the cell has machinery in there for transcribing this, for replicating it, and you can make a lot of RNA now, and that gets translated into the proteins that make the virus. You get virus assembly, the cell breaks, and that's called lytic cycle, because it's lysing or breaking the cell. So that's when the virus essentially goes viral, so to speak. That's how we use the word go viral, right? That wasn't, by the way, a word made up for the internet. It comes from this stuff. So it's going viral. It's basically making tons of itself and bursting out, okay? That's what it means. Okay, the other possibility is it can just hang out in stealth mode. So it's still in the bacterial genome. That gets replicated as part of the normal cell cycle. There's now 2 genomes. The cell divides. The virus is reproducing either way. Okay. But it can switch between these 2 states. And it's that switch that we're going to be talking about today. So here's the viral genome, and the 2 genes we're going to be interested in are CLEAR1 and CRO. And these respectively make proteins that stabilize the switch in either direction. CLEAR1 encodes a repressor protein. It looks a bit like this, not literally, of course, but schematically a little bit like this. There's the DNA binding domain, because it's a transcription factor. It's got a DNA binding domain. And it's got a bunch of different protein interaction domains. Up at the top, it's got domains to make dimers and tetramers. So it's going to, just like the lac repressor, it's going to make a tetramer. And then, unlike the lac repressor, it actually is going to positively activate transcription, and it's going to do that through an activation domain that interacts with RNA polymerase. So, like the lac repressor, it can bind to DNA. It can bind as a dimer, or it can bind as a tetramer. Let's not really worry so much about that. The configuration we're going to be interested in is tetramer binding. This is where the magic happens. There's the CLEAR1 gene, there's the CRO gene, and there are the promoters, right where you'd expect them to be. And there are 3 operators that sit between them. The CLEAR1 gene makes the lambda repressor protein, and when this gene is being expressed, you're in the lysogenic state. That's the quiet state where it's in stealth mode. When CRO is being expressed, you go lytic. Okay? That's when it makes a ton of viruses and breaks the cell. So it's the switch between these 2 that we're going to be interested in. These operators sit between the 2 genes, and the genes are transcribed in opposite directions. That means the template strand for CRO is the coding strand for CLEAR1, and the template strand for CLEAR1 is the coding strand for CRO. Okay? Polymerase is going to bind here and go that way, or it can bind here and go that way. And when it's going one way, it's using one strand of DNA as a template. When it's going the other way, it's using the other strand of DNA as a template. Okay? It has to. Okay? Otherwise, if they were going in the same direction, they'd be using the same strand as the template. Because RNA polymerase moves in a polarity. It can't go in either direction on the DNA template. It always goes in the same direction. Okay, so these operators are crucial to the switch. And this is basically how it works. When CLEAR1 is being transcribed, you make the repressor protein. The repressor makes a tetramer, and it binds to operator 2 and 1. Now when it does that, the polymerase can't attach and transcribe CRO. But operator 3 is opened up, and the polymerase can attach, and there's a promoter there for CLEAR1, and it can go in that direction. The other state is when CRO is being transcribed. There's no lambda repressor present. The polymerase can attach here and make CRO, and CRO actually makes a transcription factor that's an activator for itself. It binds to the operator number 3. That helps the polymerase bind. So this is a positive feedback cycle. Regulates its own transcription. Okay, so that's the switch. How is it thrown? Okay, so before we get there, I just want you to think about why it's essential for the way that this switch works that clear1 and CRO are on opposite strands of DNA. Why is it essential for clear1 and CRO to reside on different strands of DNA for the switch to work properly?
The oncoming is the blue form. So that the probe still can move for another system. Yeah. Yeah, I talked to her. What do you think? With all your knowledge.
I think they're on opposite strands. They're on
all
different
those
strands.
things.
They're all running in each other. Basically, like in order to be regulated at the same time, like, you don't want them both running at the same time.
But they could be. RNA bloomers.
But they're like activated by like activated or repressed, like by the same mechanism. So like the same mechanism activates one and touches the other. It's like
All right.
both your feet.
I think we have a winner.
Gas and
Because if they weren't on two different strands of the same operators could affect both genes. Right, okay, so the operator regions sit between the two genes. A single operator can influence both genes, depending on what's bound there. It couldn't do that if they were pointed in the same direction, because it would only be upstream of one of those genes. But having the genes point in opposite directions means you can have the same operators influencing the expression of both genes at once. Yep. So remember what orients the direction of the polymerase is really going to be the promoter sequence, not the operator sequence. Right, so we're going to go through I'll go through the mechanisms now. Because it's a little bit confusing because it's lambda repressor is the repressor of CRO, but it's also the activator of clear1. So let's see how that works. Okay, so this is the state where clear1 is being transcribed. There's the repressor protein, and it is repressing transcription of CRO just the way that the LAC repressor was repressing the LAC operon. It's just basically getting in the way. Now it's not sitting between the promoter and the transcription unit. It's not doing it that way, but it's just physically too big, and the polymerase complex can't come down and sit on the promoter. So it's repressing CRO's transcription, but it's also activating clear1's transcription. And remember, there's this activation domain here. right there, is helping to stabilize the interaction of RNA polymerase when you operate with the promoter region. So that's a stabilized interaction that now allows clear1 to be transcribed, which means you're making more of this, and so that's a positive feedback cycle. So it's maintaining its own transcription. So that's a stable state. Once you switch into the lysogenic state, it's going to maintain itself. Okay. Because it's a positive feedback cycle. Correct. And the other one's positive too. It's just stabilizing the opposite state. That's exactly right. Okay. So it's both an activator and a repressor. Does everybody see how that works? It's functioning in both. You couldn't do that if the two genes weren't facing opposite directions, because it requires having these operators sit between them. And here's the opposite state. Okay? So when the Cro protein is bound, I'm sorry, when the Cro protein is bound, it helps recruit RNA polymerase onto the other promoter, and you get production of the Cro gene, and so this is a positive feedback cycle too. So that's one of the things that Cro does, is it positively autoregulates its own transcription. That's helping maintain this other stable state, and it's also inhibiting transcription of clear1 because the polymerase can no longer bind over here. Okay, so each of them maintains its own activity and locks out the other state. The other thing that Cro does is it activates transcription of genes that are necessary for the lytic cycle. So once you flip into this state, you then start to produce the proteins that are going to cause lysis and the replication of the viral proteins that build the capsids, the shell of the virus, and replicate its gene. Yeah. Between the two states. I'm not actually going to talk about that today. I really want you to think about the logic of how the switch actually is executed. So the things to notice here are that this is that you lock in one system or you lock in the other through positive feedback cycles while you're at the same time negatively regulating the other one. Okay? And so they're mutually exclusive states. Okay? They're positive for their own expression, and they're negative for the other expression. Okay? That's really all I want you to get out of that. There's a whole lot more to be learned about the lambda switch. In fact, your textbook has a little bit more, but there's a whole book you can get about this called A Genetic Switch. It's a classic study in how gene regulation works. I want to end with one final example, the ribosomal operons of E. coli. And this switch works a little bit different. Because we're talking about now a switch between on and like super duper on. So it's a different kind of switch. So just to remind you, this is something you probably already know, or maybe you've learned it once and forgotten. There's 3 different kinds of RNAs that you need to make a protein. We mostly have been concerned up till now with the messenger RNAs because that's what codes the protein. But you need ribosomal RNA to make the ribosome, and you need transfer RNA to bring the amino acids during translation. So Steve's going to talk a lot about these guys later. Just be aware that protein synthesis is an extremely demanding part of what a cell does. There are a lot of ribosomes in a cell at any one moment, a lot of tRNA. So just to give you a sense in mass how much we're talking about. This is the percent of an E. coli cell. Messenger RNA is only about 2% of the cell. There are about 2,500 messenger RNAs in an E. coli cell at any one time. Of course, you're always making more and degrading, so that's turning over, but about 2,500 molecules. In contrast, there are about 160,000 transfer RNAs, and there are about 20,000 complete ribosomes. The ribosome's a big object. That's about 30% of the cell mass. So there are tons of ribosomes. If an E. coli cell divides every 20 minutes, you need to make 20,000 every 20 minutes. That's 1,000 ribosomes a minute. That's cranking. So how do you do that? That's not a normal gene. Okay, most genes you don't make anything close to that much. So for an E. coli cell to be actively dividing, it's got to be producing beaucoup ribosomal RNA. All right. So this is a very special kind of promoter. Okay. And so this picture is actually of a ribosomal operon, and you can see it's just, you almost can't get another RNA in there, RNA polymerase in there before, as soon as one gets out of initiation into elongation, you're loading another one. And as soon as it clears, you're loading another one. It's pretty much running literally as fast as it can possibly go. You can't get transcription faster than this. Okay. So how does that work? There are two promoters on the ribosomal operon, a P1 promoter and a P2 promoter. P2 is basically on. It's when the cell's not actively dividing. You still make ribosomes because they turn over, they get bad, the cell degrades them, and you make new ones. But when the cell's dividing, it's super on, and then it uses the P1 promoter. Okay, so this is a switch between on and really on. Okay, so not surprisingly, you should now know that the P1 promoter is going to use sigma 70 in its -10, -35 box binding, and the P2 is going to use a different sigma factor, which is the one that goes with very little cell growth. So remember we talked earlier about switching between sigma factors depending on whether the cell's dividing rapidly or just not dividing or maybe dividing very slowly. So that's the switch we're going to be talking about. The switch itself is pretty much executed by the presence of whichever sigma factor you have present. How do you get so much transcription? We're going to now be talking mostly about P1. Both of them bind sigma factors, but they bind different sigma factors. But what makes them a lot stronger than most genes is that they have an element just upstream called the up element. If you have, the alpha subunits of the polymerase interact with these up elements, and that helps stabilize the interaction. Okay, so they're physically attached to the RNA polymerase. They're part of the polymerase complex, but they actually have a DNA binding domain at the C-terminal end. That's what CTD stands for, C-terminal domain. That actually is a DNA binding domain, and by attaching here, it stabilizes that interaction so that the polymerase doesn't fall off again before it can make an mRNA molecule. I mean, in this case, an rRNA molecule. The other factor that makes these things work well is that these sites are very close to the perfect consensus. So the sigma factors are going to bind them more strongly than many other genes. And this is not additive, it's synergistic. So each of those gets you something, but together it buys you a lot. So you can make mutations in these different regions and see how much of a difference does it make to transcription. And mutations here have an effect, mutations here have an effect, and mutations here have an effect. But if you do mutations in two places at once, you get a gigantic decrease. Okay, so if you flip that around, think of it the other way, you're getting about a 170-fold increase from one of these sites and about a 60-fold increase from the other site. You put them both together, you're getting about a 340-fold increase over just having the sigma factor bind alone. So that's turning this from just normal transcription into super transcription. Okay, any questions about that? Yeah. Yeah, so you can make mutations here and measure how much of an impact that has as well. So the experiments one does is you take your piece of DNA usually you'd start by cloning it into a plasmid just like you guys have in the lab, and then there are ways you can induce mutations. And if you induce a mutation here or here, that's going to affect sigma binding. And if you induce mutations here or here, that's going to affect the alpha 1 and alpha 2's subunit binding. And so you can pick them apart and find out how much of an impact it has if you interfere with one of those or the other. And the second thing you can understand is that by having mutations in two places at once, that's much worse than having the sum of the two independent mutations. So it's synergistic, it's not additive. And that goes back to something I talked about last week, which is this. Not all activators are equal, they can have different amounts of an impact on transcription, but sometimes having two together buys you a whole lot more than simply adding the two. It's synergistic. Okay? That's what that word means. It's not simply additive. This is a very common phenomenon in macromolecular interactions because it involves either conformational changes that help attach better, or there's cooperative binding. Yeah. It's just a binding site for a protein. And it's not, this is the promoter. So the promoter is for the polymerase. I guess you could sort of say it is because it's actually, but think of it as it's just a binding site for the C-terminal domain of the alpha subunits. The alpha subunits are part of the polymerase complex. They're always there, but they don't always interact with the DNA. Okay, so the key thing to remember here is that when you have activation, there's cooperativity or synergism that can give you much stronger impact than the two independently. So this is the ribosomal operands are just a specific example of this phenomenon. And then you can get even more with a third protein called FIS, which binds even a little bit further upstream. So there's sigma 70 bound at P1. So there'd be a -10 and a -35 right there. There are the two alpha subunits interacting with the up element. And then here's FIS binding at about -71. So this is just a ribbon diagram showing that there's the alpha subunits interacting with FIS. So the DNA is bent a little bit, they're touching each other. And all of this is helping to stabilize the interaction of the polymerase complex with the P1 promoter. So this gives you a further 3 to 8-fold increase over simply having up in the basal or core promoter. So each of these is adding on further stability and helping to get higher levels of transcription. So this is a very special promoter because of the high levels it cranks out. Any questions? Yeah. You never want to turn ribosomal synthesis off. Nothing blocks these. There's no negative regulation. It's only on or super on. All right, we'll see you on Wednesday.
I have one more.